All of an organism's characteristics arise from the presence of genes. These genes are expressed due to the creation of proteins. Today, we are going to walk through the process of creating proteins from genetic information, the DNA within a cell.
We will first discuss how DNA is transcribed into messenger RNA, or mRNA. This occurs in 3 steps: initiation, elongation, and termination. First, transcription must be initiated. Transcription factors, proteins, bind to DNA at the TATA box, a specific nucleotide sequence upstream from the start point. RNA polymerase II, an enzyme that separates the 2 strands of DNA apart and joins RNA nucleotides along the mRNA strand, binds to the promoter region of the DNA at the TATA box, a specific nucleotide sequence at the site of the transcription initiation complex of proteins that assist in transcription. The transcription initiation complex is then formed when additional transcription factors bind to the DNA with RNA polymerase II. The DNA strands unwind in the strand running in the 3' to 5' direction, because transcription must occur, and the 5' to 3' direction becomes the template strand for the transcription of mRNA.
The next step is elongation. The transcription start point within the promoter is where mRNA formation begins. RNA polymerase untwists the double helix of DNA as it moves downstream, exposing about 10 to 20 DNA bases at a time for RNA nucleotides to be paired with in a complementary fashion. C pairs with G, G pairs with C, T pairs with A, and A pairs with U because RNA has uracil and not thymine. As the process continues and the RNA polymerase continues moving downstream, the newly formed RNA separates from the DNA, and the DNA reforms in its double helix. It is important to note that many RNA polymerase molecules can attach to a section of DNA and transcribe RNA at the same time by following each other down the molecule. RNA comes off of each polymerase. This creates more mRNA faster, making a protein in larger amounts.
Finally, the transcription must be terminated. Termination is different for bacteria and eukaryotes. In bacteria, transcription occurs until the terminator sequence is reached on the DNA template strand. When the corresponding RNA sequence is created, it acts as a signal for termination. The polymerase is detached and the RNA transcript separates from the sequence, which can immediately function as an mRNA in the next steps of protein synthesis. In eukaryotes, termination occurs when RNA polymerase II reaches a polyadenylation signal in the DNA. This signal is transcribed to an AAU-AAA sequence in the pre-mRNA that is being coded. 10 to 35 nucleotides downstream from the signal, proteins once associated with the pre-mRNA release the signal from the pre-mRNA molecule, which separates the final mRNA in the stop signal. RNA continues to be transcribed by the polymerase after this point. Research on yeast has shown that this RNA is possibly digested by an enzyme moving along the RNA strand, and that when the enzyme reaches the polymerase, transcription stops and the polymerase disassociates. Transcription is now complete.
In eukaryotes, mRNA must be modified before it can be transported outside of the nucleus and used to create amino acid sequences. First, a guanine nucleotide called a 5' cap is added to the 5' end of the pre-mRNA strand after the first 20 to 40 nucleotides have been transcribed. This helps keep the pre-mRNA protected and will eventually allow the mRNA to attach to the ribosome. At the 3' end, a 3' poly A tail will be added. The poly A tail is made up of 50 to 250 adenine nucleotides. These are added to the RNA strand by an additional enzyme. This addition stabilizes the mRNA strand and helps it to be transported outside of the nucleus for protein production. There are also regions called UTRs, untranslated regions, at the 5' and 3' end. These are part of the mRNA but will not contribute to protein formation. They help with other functions, like ribosome binding.
The mRNA is further modified before it leaves the nucleus of the cell. Pieces of the pre-mRNA, called introns, are removed in a process called mRNA splicing. Introns are segments of the RNA that do not code for proteins. They are interspersed between exons, which are expressed, in most cases through amino acid translation/protein creation. First, small nuclear nucleoproteins, consisting of small nuclear RNA and protein molecules, are able to recognize splicing sites, short nucleotide sequences at the ends of introns. These SNRPs form a spliceosome at the splicing site with other proteins. The snRNA in the spliceosome is then able to pair up with corresponding nucleotides along the intron. The spliceosome breaks the intron off of the pre-mRNA strand and joins together the exons. As the spliceosome disassembles, the mRNA with only exons is released. The mRNA is now ready to be transported out of the nucleus and used in the production of proteins.
Translation is the process in which the modified mRNA is converted to a polypeptide chain. The first step is initiation. First, the small ribosomal subunit binds to the 5' end of the mRNA strand. The large ribosomal subunit then binds to the small subunit. Both subunits of the ribosome are made of ribosomal RNA, otherwise known as rRNA. Transcription begins when the start codon, the codon AUG on the mRNA strand, is reached. At the start codon, an initiator tRNA binds mRNA strands with hydrogen bonds. Proteins called initiation factors are used to bring all of these pieces together in the initiation complex. This process of coming together requires the phosphorylation of one GTP molecule. At the end of initiation, the initiator tRNA is in the P site of the ribosome, and the A site is open for an amino acid tRNA to come along. The polypeptide is then ready to be synthesized from the N-terminus to the C-terminus, methionine to carboxyl ends.
Next is elongation. One by one, amino acids are added to the polypeptide chain by elongation factors, a type of protein. GTP, a form of energy, is used in codon recognition and the translocation/movement of tRNA from the A site to the P site and the P site to the E site. The A site is the position on the ribosome where the tRNA anticodon binds to the mRNA codon. The P site is the position in the ribosome where the amino acid of the tRNA strand is attached to the polypeptide chain. The E site of the ribosome is where the tRNA anticodon without an amino acid falls off the mRNA strand. The three steps to elongation are: the anticodon of an incoming tRNA pairs with a corresponding codon on the mRNA in the A site of the ribosome. By using a GTP molecule, accuracy and efficiency of this process is increased. An rRNA molecule located within the large ribosomal subunit catalyzes the formation of a peptide bond between the amino acid that has just arrived in the A site of the ribosome and the carboxyl end of the polypeptide in the P site. 3. The tRNA in the A site moves from the A site to the P site, while the now empty P site tRNA moves to the E site and is let out of the ribosome using one more GTP molecule. mRNA also moves in this process, exposing a new codon in the A site.
The final step is termination. Elongation ceases when there is a stop codon of the mRNA strand located in the A site of the ribosome. The stop codons are UAG, UAA, and UGA. A protein called the release factor binds to this stop codon. A water molecule instead of an amino acid is added to the polypeptide chain. The release factor breaks tRNA in the P site and breaks the polypeptide bond, so the polypeptide can move out through the exit tunnel in the large ribosomal subunit. The rest of the translation assembly is dismantled, and other protein factors help facilitate this process, which requires 2 more GTP molecules. Multiple polyribosomes move down the mRNA strand at the same time, making many copies of the same protein at once.
Now we have a functional polypeptide chain, which is folded into a functional protein.